This is Module 6, Eukaryotic Gene Regulation. In this video 6-2, Regulating Genes by Chromatin Remodeling, I'll be discussing some of the different ways that the cell repackages DNA and alters how DNA is packaged in order to change gene expression. So one of the major ways in which eukaryotes regulate their gene expression is through degradation of messenger RNA. Another major, which was discussed in video 6-1. Another major way in which eukaryotes change gene expression is through changes in the chromatin structure of DNA. Remember that DNA in eukaryotes is packaged with histone proteins and condensed into a material called chromatin, where DNA is tightly associated with proteins. If this is the case, then RNA polymerase, which is responsible for using DNA as a template to make RNA, cannot access promoters if DNA is packaged very tightly around the histones. It simply cannot bind it. There's something else already bound there. And so there's different ways to change the structure or change how tightly associated proteins are or histones are with their DNA targets. And by changing that tight association to maybe a more loose one, you can increase gene expression. Or if you make it a tighter association, you can decrease gene expression. So 3 of the methods that I'm going to talk about today, and there's one additional one as well, is chromatin remodeling and histone modification. And so these are 2 different ways to change chromatin structure by altering the proteins that the DNA is wrapped around. Okay, so first let's talk about chromatin remodeling. Chromatin remodeling is achieved by a complex of proteins and use of a single ATP molecule to basically reposition histone proteins on the DNA strand to expose different areas for transcription. So here we have up here a bundle of nucleosomes, and the sphere in the center are the histone core, and the spirals are the DNA wrapped around these these histones. And so we call them nucleosomes. In order to express genes, say, wrapped around this histone protein, you need to expose the promoter. So the first thing that happens is the chromatin remodeling complex, which is a complex of proteins and ATP, will bind to a DNA target and sort of move the nucleosomes around, so pushes them around to expose or completely unwraps one histone protein from the DNA. And this exposes transcription sites for binding. And so this could be the initiation factor for RNA polymerase, for example. And so if that is exposed, the transcription factor that helps to initiate transcription will bind, and that will stabilize RNA polymerase at that site, and then you get gene expression. In order to reverse this, the chromatin remodeling complex will come off, and then histones will be wrapped, or DNA will be wrapped back around those histone proteins. And so this represents one of the unique characteristics of eukaryotic gene expression, or one of the things that has to be overcome, which is eukaryotes package their DNA with proteins, and that makes it inaccessible to the enzymes that transcribe it. So in order to transcribe a gene and express that gene, you need to unravel the DNA in order to expose it. This is not something that prokaryotes have to worry about, since they don't package their DNA this way. Another way in which we can alter histone properties are by methylation of the histone tails. So these histone tails are small fragments of the histone core. This is the H1 histone protein that hangs off from the histone core and sort of pinches the DNA onto the histone core, sort of like you would take a piece of tape to, so if you were taking, say you had a piece of thread and you wrapped that piece of thread around a center spool, you're going to have a piece of thread sort of hanging off, and it runs the risk of unraveling. That thread is the DNA and the spool is the histone protein. And so if you want to prevent the thread from unraveling, you could take a piece of tape and tape the thread to the spool. And that's essentially what this histone tail is doing. It's sort of like a little bracket that keeps the DNA from unspooling on the core, which would be the histone core. If you methylate the histone tails, so add a methyl group like a CH3 group to these histone tails, you can actually pack the nucleosomes more tightly together. And so that makes certain DNA inaccessible to transcription factors and RNA polymerase and reduces gene expression. And so methylation of histone tails causes DNA and protein to more tightly associate, and that means gene expression will decrease. Now, another thing that you could do, so see this H1, this is the histone, one of the histone tails. Okay, and then we have some others here too. Okay, so the acetylation of histone proteins is another way to alter chromatin structure, only it has the opposite effect. And so, again, these histone tails are sort of keeping the DNA from falling off the spool, and H1 protein also helps to do this. And so not just H1 can be methylated or acetylated. Each of the histone proteins in the core also has a tail that's sort of keeping the DNA attached to the core. So you can add acetyl groups to these histone tails to have the opposite effect of the methyl groups. And so adding a methyl group to the histone tail results in more tight association between histones and DNA. Adding an acetyl group to the histone tail, however, will neutralize the positive charge on the tail and actually produce a looser association between the histone core and the DNA. And so you can see when the tails are neutralized by these acetyl groups, they are not as able to tightly associate. They can't snap or keep the DNA spooled on its spool. And this weakens the DNA interaction with the histone and will allow for transcription factors in RNA polymerase to bind to the access DNA. So those are different ways in which you can alter histone or modify histones to change gene expression. We can also directly modify the DNA in order to change gene expression through the use of methylation. Methylation is when you add a methyl group specifically to cytosine residues in a DNA polynucleotide. And so here is our DNA strand, our double DNA strand. We have the one strand on the bottom strand down here. And if you add methyl groups and you highly methylate certain areas, then that will reduce gene expression. So think of it this way. You're basically adding a bunch of tiny boulders to this DNA. And if you add a bunch of these bulky groups to the DNA, the polymerases and transcription factors that need to access it will bounce right off. They can't actually access the DNA anymore. And so some regions called CPG islands are areas of DNA where there's lots of CG pairs occurring about every 10 bases. And because cytosine is the only base that gets methylated, these areas, sometimes called CPG islands, can be heavily methylated regions and represent regions of DNA that can be regulated. And so if we have unmethylated CPG, as is shown by these open circles, then transcription will be allowed to proceed. However, if you methylate certain areas quite heavily, or you methylate these CPG islands, then transcription factors and polymerases will bounce off of those areas and you will not get any gene expression. So this means methylating DNA and methylating histone proteins will decrease gene expression. Chromatin remodeling and acetylation of histone tails will increase gene expression.